Hello, my name is Chase Patton, and today I will be summarizing an article written on the 2nd of August 2017 by Heidi Ledford and posted to nature.com. The title of the article is CRISPR Fixes Disease Gene in Viable Human Embryos, Gene Editing Experiment Pushes Scientific and Ethical Boundaries. The article goes on to summarize that a Dr. Shukrat Mitalapov led a team of researchers in using a new method of CRISPR, known specifically as Cas9, to target a gene known as MYBPC3. This gene has a tendency to cause the muscles around your heart to thicken, which can lead to a medical condition known as hypertrophic cardiomyopathy, which can be a leading cause of death for young athletes worldwide. The article continues to explain that during the process, 9 out of 10 embryos were viable, leading that the experiment was a success. But with all things leading to genetic engineering, the fears of designer babies usually comes up. The article goes on to quote a Robert Lovell badge from Francis Crick Institute in London, and I quote, "Mitalapov's team provided a strand of DNA to serve as a template for rewriting the disease-causing mutation. But surprisingly, the embryos did not use the template the researchers provided. Instead, the embryos used the mother's DNA as a guide to repair the MYBPC3 mutation carried by the father's sperm. This isn't a clear step towards a designer baby," says Lovell Badge. "This suggests that you couldn't add anything that wasn't already there." Now, as to where this article corresponds to our curriculum that we have been learning over the last 3 months, it specifically refers to Chapter 10, Biotechnology. More specifically, Chapter 10.2. In Chapter 10.2, they go on to define genetic gene therapy and genetic diagnosis. Genetic diagnosis being a process used by scientists and medical experts to diagnose a detected genetic disease. An example of this being when they try to detect for the BRCA gene. This gene is responsible for genetic predisposition to certain types of cancers, more specifically breast and ovarian cancers found in women. By locating in this gene, they hope in the future to be able to use methods such as what Dr. Mitalapov used to remove the gene and therefore provide a more disease-free and cancer-free life for human beings in the future. So, I've been Chase Patton, and that was my summary of the article CRISPR Fixes Disease Genes in Viable Human Embryos. If you want to know more or check out the article yourself, you can find it on nature.com at the URL right here. And as a final end to the video, I'd like to put out a special thank you to Anzia Magazine here in Ocala. They were kind enough to allow me to use the video equipment and audio equipment used in this video, as well as edit the video and teach me how that is done. So I got to learn a few things, besides just biology stuff. You can also find out them at www.anziamagazine.com, which is also right here. And I have been Chase Patton, and this has been my video summary. Thank you.